Hello, I am Magali Hennion and I work in the lab Epigenetics and Sulfate in Paris. I'm responsible for the bioinformatics and biostatistics platform, and I will present a workflow to analyze repeat expression that we started 2 years ago with the M1 student Ali Amraoui. So repeated elements constitute huge fractions of many genomes, with about half of the sequence consisting of repetitive element in humans. And a big part of these sequences are transcribed. And transcription misregulation is associated with several diseases, including rare genetic diseases and cancer. In most RNA-seq analysis, they are ignored. But some tools have been developed to look at their transcriptions. So here we implemented 2 approaches in a complete analysis workflow that is coded with SNCMEC, and the workflow is made to be used by biologists with no or little bioinformatics background. So first of all, why is it important to use a workflow manager? In complex analysis, there are tasks that are done for each samples. Some that combine the information from several samples, and among those tasks, some can run in parallel and some have to wait for others to complete before running. So doing those analyses manually is really time consuming and can lead to errors, and moreover it is very hard to reproduce analysis done this way. And this is not doable if you don't have a strong bioinformatics background. So we developed this RNA-seq analysis workflow in the lab, so to allow for all biologists to analyze their data on HPC clusters. So it is based on an existing SNCMEC workflow called RASflow, that was heavily modified to be more efficient and to include repeat expression analysis. Here is the structure of the workflow. So we started by either personal data, so the FASTQ files from Illumina sequencing, or from public data that will be automatically downloaded from NCBI SRA database. Then there is a classical quality control of the data and facultative trimming of the FASTQ files. So for gene analysis, it is possible to use transcript quantification using Salmon. But here I will focus on repeat expression, and for that we have to map the read on the reference genome, and it can be done with either HiSET2 or STAR. Then the feature counting can be done by feature count star HT-seq for genes, and by feature count or T transcripts for the repeats. After that, the differential expression analysis can be done for the genes and for the repeats. So the configuration of the workflow by the user is easy, using a simple YAML file where you give the path to the data, the tools to use, the different parameters, and the table that lists the samples and the group they belong to for the differential analysis. The workflow can work on different clusters using Slurm. And for instance, the ones from the IFB network presented here. And it is fast. It takes less than 2 hours to complete the workflow from FASTQ files to the final reports for complete data sets. It outputs different files for visualization in IGV, for instance, as well as a comprehensive HTML report that is easy to share between colleagues. And these reports include interactive plots, which are very useful to go deeper into the results and make new hypotheses. Focusing more on the repeat analysis, 2 algorithms are implemented. So one using feature counts that assign multi-maples randomly, while the second is based on T transcripts, and it reassigns the multi-maples reiteratively and put more reads on the repeats that already have reads using SNP information. Here is the correlation of repeat expression differences found in a dataset from human embryonic stem cells before and after in vitro differentiation. And you can see the two methods with different parameters. We can see that assigning randomly multi-mappers or discarding them do not affect a lot the differential expression analysis. On the contrary, keeping the reads overlapping two or more features makes a huge difference, and this is necessary to get results with feature counts that are close to the ones obtained with the TE transcript method. As a general conclusion, we can say that the choice of the method influences a lot the results at the scale of a single repeat. But if you look at classes or families of repeats, which is generally sufficient, the results are much more stable. We're still working with biologists to add figures to sum up repeat expression results in a more comprehensive way. And finally, I'd like to thank Ali Amraoui for his work on the workflow, Jean-François Ouimet and Guillaume Velasco and Madeleine Muscatelli for discussions and data, Olivier Kirsch and BIB's steering committee, Julien Rey for maintaining our local HPC cluster, and the Société Française de Bioinformatique for its financial support that allowed me to attend this meeting. Thank you.